The G1 phase is part of interphase of the cell cycle. A cell begins to grow in size and it performs its normal task. This phase is responsible for checking the preparation of the cell for DNA replication. In the S phase, the cell's generating material, it duplicates in order for the offspring of the two new formed cells in mitosis phase can receive both an equal number of genetic material. This phase then proceeds to check the readiness of the cell for the G2 phase. The G2 phase is where the organism makes sure that any new cells are not defective. So the strands of DNAs have to be complete without any breaks, and there has to be a correct number of twice the strands of the original cell. If the cell finds a break, the DNA strand is repaired. So during the G2 phase, extra protein is often synthesized and the new organelles multiply until there is enough for the two cells. Prophase is the first phase of mitosis. This process separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. So during prophase, the complex of DNA and proteins that are contained in the nucleus, known as chromatin, it condenses. In metaphase, the pairs of chromosomes are all lined up in the center of the cell. These chromosomes then become visible. So during this stage, the nucleus disappears and the chromosomes appear in the cytoplasm of the cell so that they can be pulled apart into the two daughter cells in the next phase of mitosis. Telophase is the final phase of mitosis. So during telophase, a nuclear membrane forms around each set of chromosomes to separate the nuclear DNA from the cytoplasm. The chromosomes begin to uncoil, which makes them diffuse and less compact. Along with telophase, the cell undergoes a process called cytokinesis, which divides the cytoplasm of the parental cells into two daughter cells. But wait, let's go back a little. If we compare normal cells to cancer cells, they would be pretty different because cancer cells, unlike normal cells that have lower levels of dividing cells, cancer cells have large numbers of dividing cells. So instead of stopping here at the separating mitosis phase, they would keep dividing themselves